Okay, good afternoon students. In previous class, under the chapter biotechnology, principles and processes, in this particular concept, we were discussed regarding what the cloning vectors, then there we discussed the features that are required to facilitate the cloning vector, then the structure of plasmid PVR 322. Afterwards, we have discussed about the competent host for the transformation in the recombinant DNA. Okay. Now, in today's class, under the biotechnology of principles and processes, we are going to discuss the concept processes of the rDNA technology. Processes of the rDNA technology. What are the processes for the rDNA technology? Still so far we have discussed about the vectors, then we have discussed about the what we are going to call it as a tools for the rDNA technology. In that tools where we have discussed restricted enzymes, DNA ligase, then we have discussed about the plasmids. We are going to discuss the plasmids, then the E. coli bacteria, that is a host cell, okay, alien DNA, etc. Now, what are the processes? What are the steps involved in the formation of rDNA? Okay, or we are calling it recombinant DNA. And we have discussed about the we have discussed regarding the diagrammatic representation of the recombinant DNA. Recombinant DNA, we have discussed about the diagrammatic representation. Simply, we have discussed regarding what restricted enzymes, then DNA ligase, then we are going to transfer that into the host bacterial cell, E. coli bacteria, then they are going to grow in a number, then we are going to get the what? Recombinant DNA. Okay, that is a simple diagrammatic representation we were studied. And in today's class, what we are going to do, we are going to study the processes of rDNA technology in detail. What are the steps which are involved in what we are going to call that as a rDNA technology. What are the steps? Different steps are involved in a rDNA technology. The several steps are there. In that, first one we are going to go for the first step. It is the isolation of DNA. Second one is a fragmentation of DNA by the restricted endonuclease. Then isolation of desired DNA fragments, ligation of a DNA fragments into vector, transforming the recombinant DNA into the host, cutting the host cell in the medium at a large scale, and extraction of a desired product. Extraction of a desired product. So what else we used to do for getting the DNA, means rDNA or a recombinant DNA or a desired gene? First we are going to isolate the gene, isolate the DNA which you are interested. Afterwards, you are going to cut that DNA and you are going to make the fragments. Then you are going to separate those fragments with the gel electrophoresis. Then again you are going to join those cut DNA fragment to the plasmids. You are going to ready the what R plasmid, means a recombinant plasmid. Then that plasmid you are going to introduce into the E. coli bacteria. There you are going to increase the number of those bacteria, and then you are going to start extracting the rDNA from those bacteria in a large scale. Okay, these are the steps involved in processes of rDNA technology. So, in detail we are going to study these all the steps. Okay, one by one. Now I want to start with the first step, which is present in a process of rDNA technology, that is isolation of genetic material, that is the DNA. First you are going to isolate the genetic material, that is DNA. You all know the genetic material. Nucleic acids are the genetic material in the living organisms. And nucleic acids are nothing but the DNA and RNA. The DNA is represent the genetic material in most of the living organisms. Now, here in a process of rDNA technology, you are going to isolate the genetic material, that is the DNA. First thing you remember, when the DNA is present in a cell, it is not in a pure form. That is present inside the membranous nucleus, Okay, if you are going to remove that DNA, if you used to remove the DNA or extract the DNA, you used to break that particular cell, and the genes which are present, the genes are present on a long, what we are going to call as a length of a DNA, and then you are going to get several what macromolecules which are associated with this DNA, like RNAs, proteins, lipids, or anything and other things. That time, what we are going to do? You used to go for isolating the pure form of DNA. Isolating the pure form of DNA. How we are going to isolating the pure form of DNA? We are going to, let us start discussing about the isolating the genetic material as a DNA. Here, DNA has to cut. First, we used to cut the DNA with the restricted enzymes, and it is in the pure form and free from the other macromolecules. As I told you, the DNA is associated with the what other macromolecules, so that when you are going to go for the isolating the DNA, you used to cut that DNA with the restriction enzymes. Then you are going to extract that in the pure form, in the sense that is free from other macromolecules. DNA is free from other macromolecules. Now, the DNA is enclosed within the cell membrane, within the membranous. Many other membranes, cell membrane nucleus membrane. We have to break the cell and release the DNA along with, is to break the DNA or cell, and that time the DNA is going to release along with the macromolecules. That DNA is released with the along with the macromolecules. What are those macromolecules? Like RNA, proteins, polysaccharides, and lipids. What? RNA, protein, polysaccharides, and lipids. And what is the next important point is, we used to extract the DNA in the pure form. That is, the DNA is free from RNAs, proteins, polysaccharides, and lipids. Then what are the process? What are the processes are there? What is the process that can use for extracting the DNA in the pure form? That is free from RNAs, proteins, polysaccharides, and lipids. So there are some enzymes. There are some enzymes which are helpful for extracting the pure form of DNA. Okay, extracting the pure form of DNA. What are those in the sense? Lysozymes. Lysozymes, these are the enzymes in a bacteria. Using those enzymes, what are those enzymes? Lysosomes are the enzymes which are present in a bacteria. Using these enzymes, we are going to extract the genetic material in the pure form. In plant cell, cellulose enzyme which is helpful for extracting the pure form of DNA, and in fungus, chitinase that is used for the extracting the what we are going to call as a DNA in the form in the pure form. Okay. Now next thing is, I told you genes are located on a long molecule of DNA. Genes are located on a long molecule of DNA. And there you are going to find proteins such as histamines. Now here the DNA is what? We used to make free from proteins by using the protease. We used to make the DNA free from RNAs using the ribonuclease. And after this, we used to go for purify the DNA. Ultimately, what? Precipitate out after the addition of chilled ethanol. Then we are going to extract the pure form of DNA. Then you are going to treat that pure form of DNA with a chilled ethanol, and at last we are going to go for the collection of a fine threads on a suspension. Fine threads on a suspension. In this manner, you are going to extract. The pure form of DNA. First, you are going to go for the isolation of the genetic material. Isolation of the genetic material. That is DNA. The DNA, and I told you this DNA is not in the form, means free form. That is isolated. That is associated with the other macromolecules. That is RNA, protein, polysaccharides, lipids, etc. That's what you are going to treat. That what material with a certain enzymes based upon from which you are going to extract it. You are extracting from the bacteria. You are extracting from the plant cell, or you are extracting from the fungus. We used to use the different enzymes like lysozyme, cellulose, chitinase. Then we are going to make DNA free from the proteins. We used to use a protease. If we used to make the DNA free from the RNA, you use the ribonuclease. After that you are going to get the pure form of DNA. Then you are going to treat that pure form of DNA with the chilled ethanol, and at last you are going to collect the DNA collection of the fine threads in a suspension. Fine threads like a suspension. Okay, this is about what you are going to extract or isolate the DNA in a thread-like structures. So next step, what you are going to go for after the isolating this, you are going to go for the cutting of DNA at specific location. And second step is, after the isolation of this DNA, you are going to go for a cutting of that DNA at a specific location. That is very important, and I am not going to write each point regarding that. You used to recollect the points and write the notes for this. What is the second point? I used to explain that here. Cutting of DNA at specific location. Cutting of DNA at specific location. Cutting of DNA at specific location. Okay. So now it is a job to cut the DNA at a specific location, in the sense at a interested gene. There are several genes which are present on a length of the DNA. You used to cut that DNA at a specific position. How you are going to cut the DNA in a specific position? You all know that restricted enzymes are used as a molecular scissors. We are going to, there are the two kinds of restriction enzymes, endonuclease and exonuclease. They are going to cut the DNA within the sites, and other type of endonuclease, they are going to cut the DNA at the end. Okay, so here in this particular point, cutting of the DNA at a specific location, in the sense restriction enzymes, digestion are performed by the incubating the purified DNA molecule with the restriction enzyme. What we are going to do here, first, whatever you are taken pure form of DNA, okay, from the isolating the genetic material, first step, we are going to incubate this. Incubating this pure form of DNA with the restricted enzymes. We are going to incubating this with the restriction enzyme at the optimum condition for that specific enzyme. This is what? At optimum conditions which are required for the enzymes. What we are going to go for first? Cutting of the DNA at a specific location. We are going to take the isolated pure form of DNA, that incubating with the restriction enzymes at a optimum condition. Restriction enzyme at a optimum condition. Okay. Then you all know, after the cutting this for DNA, cutting the DNA, what you are going to go for? You are using the gel electrophoresis. And you are going to separate cut fragments. Alright? Separate the fragments. Okay. What you are cutting the DNA at a specific site? The purified DNA is to incubate with the restriction enzymes at an optimum condition of that particular enzyme. Then after the cutting this DNA, you are going to go for the gel electrophoresis. There you are going to separate the fragments by, you all know about the gel electrophoresis, forcing these fragments towards the anode, forcing these fragments, because the DNA is negatively charged molecule. Okay, so forcing this towards the anode, you are going to start separating the fragments. Then what we are going to go for, and you all know about the processes in a gel electrophoresis, in the sense agar gel you are going to use the matrix and you are going to apply the electric force. That time the DNA is a negatively charged molecule. The fragments are start moving towards the anode. The bands are going to obtain on a agar gel. Then you are going to go for a what I told you regarding what staining with the ethidium bromide. After the staining with the ethidium bromide, you are going to visible. Then you are going to cut and separate. That is what elution. Okay, it is come under the electrophoresis. In this particular step, you are going to write this much only: the cutting of DNA at a specific location. The pure form of DNA, it is incubated with the restriction enzymes at an optimum condition. Then the fragments are separated by using the gel electrophoresis. That is what agar gel. There the DNA are negative charge and move towards the anode, and we are going to extract it out by the process of elution. Then after the separating these, what we are going to call as the particular DNA fragments. We have to rejoin them. We have to rejoin them to the plasmid. You have to rejoin them to the plasmid or a what we are going to call it as a vector. We used to rejoin them to the vector. The interested gene is rejoined to the vector with the help of what? DNA. With the help of DNA ligase. What I told you here, after having the cut source of DNA as well as the vector with the specific restricted enzyme, the gene interested from a source of DNA and the cut vector is mixed with the what? Vector with the help of ligase, L-I-G-A-S-E, ligase. Okay, and the result is the preparation of recombinant DNA. After that, we are going to go for the preparation of the result is to come, that is the rDNA in a vector. Whatever that vector, the plasmid or a bacteriophage. So, in simple, I will tell you, first isolation of the genetic material used to isolate the genetic material DNA in the pure form, because that DNA is associated with the macromolecules like RNA, proteins, then polysaccharides, lipids, etc. And you used to treat with the different enzymes, and then you are going to purify that, and you are going to treat the DNA with the chilled ethanol, and you are going to get the precipitate, the thread-like structures in a suspension. Afterwards, you are going to go for the cutting of the DNA at a specific location. Here, the isolated DNA, pure form of DNA threads are incubating with the restricted enzymes at a optimum condition. Then the DNA is going to cut at a particular position. Then you are going to separate those cut end of a fragments with the help of gel electrophoresis. Okay, and afterwards, whatever you are going to cut the DNA fragments, those DNA fragments has to rejoin. In the sense, you are going to cut the vector. For example, plasmid. Now, very good diagrammatic representation of the rDNA. Both you have to cut with the restricted enzymes, the alien DNA. Then you are going to cut the plasmid or a vector. Then afterwards you used to rejoin them. Interested gene is inserted into the vector. For example, a plasmid with the help of DNA ligase, and that plasmid is ready as a rDNA for the next process in a process of rDNA technology. Okay, so these are the two first steps which are involved in the processes of rDNA technology. The next used to come, that is the amplification of gene of interest by using the PCR. PCR in the sense polymer chain reaction. I am going to continue that what amplification of the interested gene in the sense rDNA ಏನು ಮಾಡಿದ್ವಿ ನಾವು ಯಾವುದು ನಮಗೆ ಇಂಟ್ರೆಸ್ಟೆಡ್ ಐತಿ ಅದನ್ನ ಏನು ಮಾಡ್ತಾನೆ amplification ಮಾಡಬೇಕು. amplification in the sense you are going to increase in a number that particular what gene. And next step, insertion of recombination and obtaining the origin gene product, that we are going to continue in a next class. The present class we have discussed these two steps which are involved in a process of rDNA technology. One is isolation of genetic material, pure form of DNA, and cutting of the DNA at a specific location. Here in this step we are going to remember the different kinds of enzymes now, and in this second step we are going to write the pure form of DNA incubating with the restricted enzymes at the optimum condition, then it has to cut the DNA at a specific sites. Then you are going to go for the gel electrophoresis by using the what agar gel and electric field. You used to what force the DNA is moved towards the anode because the DNA is negatively charged. Afterwards, the DNA fragments used to rejoin to the vector, for example a plasmid. That is what with the help of DNA ligase, and that is ready as a rDNA, a vector or for the further. And next we are going to amplify the interested gene and the collection of the interested gene, that we are going to continue in a next class. Okay, I will stop here. Thank you.